Swayam Prabha. Digital India. Educated India.
Welcome to today's lecture, which is on biomolecules. As I have already talked to you about the cell, and I have already mentioned that cells are the basic component of any living system. And when I talked to you about the cells and its different organelles, I have also told you about the fluid which is present inside the living cell. And this fluid is called the protoplasmic fluid. And here some macro and micromolecules are present. And today I'll be talking to those type of micro and macromolecules. So my today's lecture is mainly on the biomolecules which are present in any living cell. Now if we see the biomolecules, it mainly made up of the carbon, hydrogen, nitrogen and oxygen. Now if we see these macromolecules which are present in any living cell, we can categorize them into proteins, lipids and carbohydrate. Now today my lecture will be mainly focused on the carbohydrate molecule. Now when I have told you that these biological molecules are mainly made up of carbon, hydrogen, nitrogen and oxygen, and when we are talking about carbohydrate, this carbohydrate moieties, in this carbohydrate molecules no nitrogen atoms are there. Nitrogen molecules are not present in this carbohydrate molecule. If we see the empirical formula of any carbohydrate, then we can find that it is roughly about CH2O to the power N, when N is the number of carbon atom which is present in any carbohydrate molecule. That means if we see this formula, it appears that it is a carbon hydrated form. Now carbohydrates are the polyhydroxy of organic compounds which contain the aldehyde and ketone. That means that CHO, the aldehyde group, or C11O, that keto groups are present in the carbohydrate molecule. Now there are four major class of carbohydrates which can be distinguished as monosaccharide, disaccharide, oligosaccharides and polysaccharide. Now while talking to these monosaccharides, this monosaccharide is a unit sugar molecule. That means it is a single sugar which is present in any carbohydrate. Now when we are talking about the disaccharide, it is the two sugar moieties which are joined together. That is the disaccharide. When we are talking about the sugar molecules which varies its chain length from 3 to 10 units, that is called oligosaccharides, and when the number of sugar moieties varies from 100 or 1000 units, we call those type of sugar molecules as polysaccharide. That means from unit a single sugar molecule we can get a long chain sugar moieties. This, if we see this monosaccharides are the colorless crystalline solids that are freely soluble in water but insoluble in non-polar solvent. Both it can be obtained, it can be found in both open chain and ring structure, and also we can get this monosaccharide in its isomeric form. Multiple chiral carbon atom is present in these monosaccharides, and monosaccharides are generally, it starts from 3 carbon sugar to 7 carbon and so on. So, if we see the most commonly found monosaccharides, then we can categorize these monosaccharides from triose, tetrose, pentose, hexose, heptose and so on. What does it mean? Triose means it is the 3 carbon containing sugar molecule. That means glyceraldehyde, dihydroxyacetone, these are the example of 3 carbon sugar, that is the triose sugar. Whereas in tetrose, 4 carbon sugar moieties are there, like erythrose, erythrulose. In pentoses, 5 carbon sugar, the examples are ribose or xylulose. In hexoses, we are getting glucose, fructose, mannose, galactose and so on. In case of heptoses, 7 carbon containing sugar, that pseudoheptulose are the example of the simple sugar. That means these carbohydrates can be once again divided into simple sugar and the compound sugar. Simple sugar means it is a unit sugar molecule varying its carbon skeletal structure from 3 carbon to 7 carbon and so on. When we are talking about this compound sugar, then this compound sugar is once again divided into oligosaccharides and polysaccharides. In case of this oligosaccharides, it is divided into disaccharide, trisaccharides. Disaccharide means 2 sugar moieties are joined together like sucrose, maltose, lactose and so on. While talking to this trisaccharide, it is the raffinose, maltotriose, that means 3 sugar moieties are joined, linked together. While talking to this polysaccharide, that means here n number of sugar moieties are connected together. It can be divided into homopolysaccharide and heteropolysaccharide. So, homopolysaccharide, the name itself indicates that it is a single monosaccharide sugar which is present in n number of units and they form the starch, glycogen, inulin and so on. In case of heteropolysaccharide, there are plenty of examples, that means along with the sugar, it is a complex molecule, that means along with the sugar, some other biomolecules are linked and it forms the different type of complex sugar structure. Say it may be the 2 sugar moieties, say for example, one pentose got linked with one hexose and in this way the units are repeated and it forms the hemicellulose. So, it is not a single pure compound, but it is the mixture of 2 different, one pentose sugar and one hexose sugar in case of hemicellulose compound. Now, besides that classification, we mainly say that in monosaccharide that there are 2 different families are there. In one group, aldehydic group is present. That means in that particular group, CHO is present. That means those type of sugars are called the aldose sugar. In another group, the ketone group is present and we call that type of monosaccharides as the ketose sugar. Besides this, these monosaccharides are also identified by its D and L form. That means it is the either D form or L form. In most of the biological cases, we mainly get the sugar moieties in D form, that means dextrorotatory. Now, when the glyceraldehyde molecule, glyceraldehyde is the simplest sugar molecule which is 3 carbon sugar, and when this glyceraldehyde exists as 2 enantiomer, first identified by their opposite rotation of plane polarized light, and when it rotates in the clockwise direction, it is denoted as plus and is designated as plus glyceraldehyde. When this enantiomer gives the opposite rotation, that is in the anti-clockwise form, it is denoted by the symbol negative, and it is the levorotatory form. The direction of rotation of light does not correlate to any structural feature. That means, how to distinguish the D form and the L form. Now, this is the structure of glyceraldehyde. Now, here this carbonyl carbon, that CHO group is present, CHO, that is the aldehydic group, as it is an aldose sugar. Now, here this is the chiral carbon. Chiral carbon means the 4 valencies are occupied by 4 different groups, and here if we see this chiral carbon which is far from this carbonyl carbon, the hydroxyl group which is present is on the right side, indicating its dextrorotatory group, that D form. Whereas in case of this structure, here you see, here the hydroxyl group is present on the left side, indicating that it is the levorotatory L form. So, this is the structure of glyceraldehyde. This is a 3 carbon sugar, CHO, CHOH and CH2OH. Here this OH is on the right hand side, indicates it is the D form, and here it is the left side, indicating the L form of glyceraldehyde. Now, the another characteristics of the monosaccharide is its isomerization. Now, in most of the monosaccharide, isomerism is a very common phenomenon. Compound with the same molecular formula but different in structural formula is the isomer to each other. Now, as I have told you that if we see the aldehydic sugar and one keto sugar, in case of aldehydic sugar, the first carbon atom which is there, it is the CHO, that aldehydic group. It occupies the first carbon atom of this skeletal structure. And if we do the numbering, 1, 2, 3, 4, 5 and 6, this is the hexose sugar. First carbon is occupied by the CHO, that is the aldehydic group, whereas in case of keto sugar, what is there? If we do the numbering, 1, 2, 3, 4, 5 and 6, we will find that carbon atom number 2 is occupied by the C11O, that ketoyl group. So, this is the D glucose. Why it is D glucose? See, the chiral carbon which is far from this carbonyl carbon, and hydroxyl group is present on the right hand side. So, this is D glucose and this is D fructose. So, this is the structure of D glucose and D fructose. Now, if we see this basic structure of glucose, then we can find that it has got the isomerization. What is that? If we see the other hexose sugar like D-mannose and D-galactose, so here this is the chiral carbon which is far from this carbonyl carbon, and hydroxyl group is present on the right hand side on each of the sugar, indicating it is the D-galactose, D-glucose, D-mannose and so on. Now, when we compare the anomeric, this isomerization, then what we will find that see the carbon atom number, this is the first carbon, this is the second carbon, and if we see the structure of this second carbon, we can find that this hydroxyl group is present. In case of glucose, it is present on the right hand side, whereas in case of mannose, it is present in the left hand side. That means it is just opposite with respect to that carbon atom number 2. That means these are the mirror image to each other, or it is the epimer. In case of the galactose and glucose, if we see the fourth carbon atom, that means 1, 2, 3 and this fourth carbon atom, we will find that this hydroxyl group is on the right hand side, whereas here the hydroxyl group is present on the left hand side, indicating that galactose and glucose are isomer to each other with respect to the carbon atom number 4. So, this is the example of aldehydic sugar. When we are coming to this keto sugar, we can find that, see here I have already mentioned that carbon atom 2 is the ketoyl group. So, here if we do the numbering, this is carbon atom 1, 2, 3, 4, 5, and that is why it is the ribose sugar, that means it is 5 carbon containing sugar. If we see the third carbon atom 3, then we can find that in case of ribulose, the hydroxyl group is present on the right hand side, whereas in case of xylulose, the hydroxyl group is present on the left hand side. That means they are isomer to each other with respect to the carbon atom number 3. And as the hydroxyl group is present on the chiral carbon far from this, the carbonyl carbon, it is the D form of sugar. Now coming to the another characteristics of this monosaccharide. Now, when we are talking about this monosaccharide, we can find that in aqueous solution, many monosaccharides, they form the ring-like structure. The formation of this ring structure is the result of a general reaction between the alcohol and the aldehyde group or the ketoyl group, which is present in the monosaccharide, and it forms the derivatives which are called either hemiacetals in case of aldehyde or hemiketal in case of keto derivative sugars. Pentoses and hexoses can form the ring-like structure as the ketone or aldehyde reacts with the distal hydroxyl group which is present, and this type of representation is called Haworth projection. Now, when we are talking about this, that means what we are talking? We are talking about the simple straight chain of glucose. Now, what they are telling that there is a tendency of this first carbon and the fifth carbon this is the tendency, it is in the ring like structure. What they are doing, they are just coming very close to each other, that if we are doing the numbering, this is the first carbon, second, third, fourth and fifth. Now, here this fifth carbon, you see this CHOH. Now, you see the structure here, this CHOH. So, here this OH and H is there and it is forming the ring like structure. Now, this is the tendency which is there commonly found in case of the monosaccharides. Now, this structure are denoted as the pyran and the furan ring. Now, what is pyran and furan? It is a heterocyclic organic compound. The 5 carbon atom and 1 oxygen atoms are present in a ring structure, and the term pyran is also often applied to the saturated ring analog, which is more properly referred to as tetrahydropyran. In this context, the monosaccharides containing a 6-membered ring structure are known as the pyranoses. Now, here if we see, this is the pyran structure, and if we see the carbon number 1, 2, 3, 4 and 5 and 6 is joined together, and this 5th carbon is getting joined in this particular pyran ring. That means, in this ring 5 carbons are present. In case of furan, we can get that 4 carbons along with the oxygen ring. That is, the monosaccharide containing a 6-member ring system are also known as the furanose. And it consists of 5-member aromatic ring with 4 carbon atom and 1 oxygen molecule in its core structure. So, if we see the anomerism, that is that pyranose and furanose form the aldehydic, that is C1, and for keto it is the carbon atom 2, carbonyl carbon becomes a new asymmetric carbon to this carbon skeletal structure. The additional stereoisomerism is called anomerism. The results of the orientation of H and OH around the new asymmetric carbon atom on the anomeric carbon which is present in this chain. The alpha anomer is the anomeric hydroxyl group which is oriented furthest from the CH2OH group, and in beta anomer the hydroxyl group is oriented close to the CH2OH group. What does it mean? It means that if this is the glucose structure, this is the first carbon which is the aldehydic group, the carbonyl carbon, and I told you that first carbon and 5th carbon, they can form the ring like structure. Now, what is happening if we see this is the carbon number 1, 2, 3, 4, 5 and 6, this 5 carbon compound, this chiral carbon, see CH and OH. This OH, here this oxygen is having the lone pair of electron which is sharing with this carbonyl carbon, and they form this ring like structure. And when they form this ring like structure, depending upon the orientation of this hydroxyl group, if this hydroxyl group is is oriented furthest from this CH2OH group, that means if this is the downwards that and far from this CH2OH group, then it is called as alpha or the trans form. And when it is oriented, this hydroxyl group is close to that of this CH2OH group, then it is the beta form, or it is called the cis form. So, alpha anomer is called as trans form, whereas beta is called the cis form. That means this is the trans and this is the cis orientation of this glucopyranose, alpha T glucopyranose and beta T glucopyranose. Similarly, in case of this D-fructose, when we are talking about, that means here this second carbon is the ketoyl carbon and here the fourth carbon has got this is the chiral carbon which is farthest from this carbonyl carbon, and OH is on the right hand side and they can form the ring like orientation. Now, if this second carbon and fourth carbon they form this ring, we call this structure, if it is like this first, second, third and fourth and fifth, sorry, this is the fifth carbon, this is first, second, third, fourth and fifth, that means second carbon and the fifth carbon, when they come close to each other, they share this lone pair of electron and form this ring structure, and it is called the alpha D fructofuranose. When it is the 5 carbon ring, that is the pyran ring, then you see this is the second carbon, third, fourth, fifth, and here the sixth carbon is coming to the picture, and here this OH group of the sixth carbon is coming and forming this ring, and it is called alpha D fructofuranose. And this is the structure, that ring formation with the glucose and the ketoyl group. Now, here you see that a 6-member pyranose ring by the reaction with C2 keto group with the OH on C6, and a 5-member furanose ring by reaction with C2 keto group with the OH on C5. So, this is the alpha D fructofuranose. What I have told you here, the same thing is that alpha D fructofuranose structure. So, this is all about the monosaccharide and its possible orientation, what we are getting in the solution form, in the ring form, and it is very commonly found. Now, coming to the disaccharide. Disaccharide means when two such sugar moieties are linked together. That means a disaccharide is a carbohydrate formed when two monosaccharides undergo a condensation reaction which involves the elimination of a small molecule such as water. That means when such type of bondings are there that release one molecule of water out of this from the functional groups only. Like monosaccharide, disaccharides also dissolved in water, it tastes sweet and are called sugar. And this is one of the example. Now, when we are talking about the glucose molecule, so if we draw the structure of So, if we are talking about the glucose molecule, and when such two glucose moieties are present, say so this is one glucose moiety and this is another glucose. Now, when this is, and if we do the numbering, and if we are doing the numbering like this, 1, 2, 3, 4, 5 and 6. Similarly, 1, 2, 3, 4, 5 and 6. So, here when these two glucose molecules are linked together, that means here if two glucose molecules are coming with each other, they form the bond with the release of one molecule of H2O. This is the linkage, and when this bonding are formed, it is called alpha 1,4 glycosidic linkage. So, this is the first carbon is getting linked with the fourth carbon of the another sugar moieties, and they are forming the glycosidic linkage. So, when one glucose molecule is getting linked with another glucose molecule, the bonding is called glycosidic bonding, and it releases a molecule of water from this bonding. So, one molecule of water is being released to stabilize this. Maltose is one of such example. Coming to the lactose, a disaccharide that occurs mainly in milk. Lactose is a reducing sugar and it exhibits mutarotation. It forms with the 1,4-beta-D galactopyranosyl and 1,D glucopyranosyl. That means, when one galactose and glucose moieties are joined together with the beta 1,4 glycosidic linkages, it is called it forms the lactose molecule. So, one galactose and one glucose is joined together with beta 1,4 glycosidic linkage, and here it is alpha 1,4 glycosidic linkage in case of maltose. So, this is alpha 1,4 glycosidic linkage, this linkage, but here what is happening, this is the beta 1,4 glycosidic linkage is formed when one galactose and one glucose molecule is joined together, and when these bonds are getting split, then we get one glucose and one galactose. Now, coming to the another disaccharide which is very interesting, and generally we use that disaccharide as a table sugar, and it is the non-reducing sugar and does not undergo mutarotation. It is connected with the acetyl form from both anomeric carbon, that is one aldehydic and one keto. So, see the structure here, when one glucose is getting linked with one fructose molecule, that 2,1 linkages is there. That means, first carbon and second carbon is getting linked with the first carbon of the glucose molecule is then it forms the sucrose molecule, and this sucrose is one of the example of the normal table sugar. Coming to this cellobiose. Cellobiose is the 2D glucopyranose unit which is joined by beta 1,4 glycosidic bond. Now earlier what I was talking to you about maltose, lactose, sucrose, those disaccharides are the sugar molecule. But here what I am talking about, the cellobiose, it is a non-sugar disaccharide molecule. Here you see one glucose is connected with another glucose moieties with beta 1,4 glycosidic linkages. It can be obtained and it can be cleaved by enzymatic method with the help of the enzyme cellobiase, and this type of compound is very commonly found in cotton, jute, paper, etc. Now coming to the polysaccharide, one of the example is the glycan molecule. The complex carbohydrate in which many simple sugars are linked, and mainly this polysaccharide and glycan. I have already talked to you about the glycan unit, if you remember, when I have talked to you about the bacterial cell structure. Then I told you that NAM and NAG, N-acetylglucosamine, N-acetylmuramic acid, those are the glycan chains which are giving the strength to the cell wall of the bacteria. Similarly, this is a polymer of the carbohydrate moieties, and here its function is that energy storage molecule, or it gives the structural support. Now if we see the polysaccharide, as I have already discussed to you, that it may be the homopolysaccharide or the heteropolysaccharide. Now coming to this homopolysaccharide, that if we are talking about this animal system, then chitin is one of the polysaccharide which is present on the exoskeleton of cockroach. Now this is the cockroach where this compound, this biomolecule is present on the exoskeleton, whereas glycogen is used as a storage food material in case of the animal system. As we have the glycogen which is the storage food system in liver. In case of plant, cellulose and starch are the example of polysaccharide. Cellulose is beta 1,4 linkage and starch is that alpha 1,4 and 1,6 glycosidic linkages. Now this cellulose, it consists of that beta 1,4 glucopyranosides of the cellobiose, and cellulose molecule form a large aggregate structure held together by the hydrogen bonds. It is the beta 1,4 linkages which are there in a linear unbranched structure, and it gives the plant the strength or the stability that it protects the plant. 100 billion tons of cellulose produced worldwide by the plant, and 50% of the earth's carbon is tied up with this biomolecule. While talking to you about the starch, I have told you starch is mainly composed of the amylose and amylopectin. Now amylose is insoluble in water and it composed mainly 20% of this starch. The alpha 1,4 glycosidic linkage is the link of glucose moieties. In case of amylopectin, it is soluble in water and contains 80% of the starch. If we see the structure of amylose, we can find that, as I have told, that alpha 1,4 glycosidic linkage is there in case of this amylose molecule, and the end of this polysaccharide with the anomeric C1 carbon that is not involved in the glycosidic bond, and that is the reason it is called the reducing end. Glucose storage in polymer form minimizes the osmotic effect, and it is a straight chain molecule, whereas in case of amylopectin it is a branched chain of this alpha 1,4 and alpha 1,6. So obviously this structure is little bit more complex than that of the amylose structure, and every 25 glucose straight chain moieties there is a branched chain of alpha 1,6 glycosidic linkage. Now when we are talking about the glycogen, glycogen is the storage food material of animal, whereas starch is the storage food material of plant. Now when we see here also the structure is alpha 1,4 and 1,6 linkages. So straight chain is the alpha 1,4 linkage, and whenever this branching is there then it is alpha 1,6 linkages. Now when we are talking about alpha 1,6 linkage, that means here then how it is different from that of starch. Here the frequency of branching is more compared to that of the starch molecule. It is a highly branched and larger than amylopectin. Up to 1 lakh glucose units are present in case of glycogen. Now when we are talking about the other carbohydrate molecule which are present in different other along with the other biomolecules. Now while talking to the nucleus, I have told you about DNA and RNA. Deoxy sugar have an OH group which is replaced by an hydrogen. That means one oxygen is missing in deoxy sugar. It is the derivatives of 2-deoxyribose and are the fundamental unit of DNA molecules, and this is the ribose sugar which is present in the RNA molecule, and these carbohydrate moieties are playing a very important role in this structure of DNA and RNA. Now besides this, the oligosaccharides of glycoprotein and glycolipids are also playing a significant role. I think if you remember, I have already mentioned during while talking to you that cell structure of bacteria. There I have told that some lipoprotein, some glycoprotein, some glycolipids are present on the cell structure of cell wall of the bacteria. So here when one glucose, that carbohydrate moieties are linked with the protein moieties or any carbohydrate moieties are linked with any lipid moieties, they form this glycoprotein or glycolipid and so on. It is making the structure little more complex. Now the hydroxyl, the OH group, in case of the amino linked amino sugar, the hydroxyl group is replaced by the amino group of the amino acids or the protein moieties. So similar way, this type of linkages are also found in case of this lipoprotein, glycoprotein and so on. So when we are talking about this amino sugar, this OH group, as I have already told you, that it is replaced by this amino group which is present, and amino sugar are found in antibiotics, say for example that gentamicin. It is also present in glucosamine. That glucosamine is one of the component of the cell wall of bacteria. So this type of complex structures are found. Now if we come to the importance of carbohydrate, then we can find that when these carbohydrates are getting oxidized, it gives the energy. And to which type of cells? The non-photosynthetic cells like us. Other carbohydrate polymers lubricate the skeletal joint and participate in recognition and adhesion between the cells. More complex carbohydrate polymers covalently attach to proteins or lipids, act as a signals that determine the intracellular location or metabolic fate of the hybrid molecule, which are otherwise known as the glycoconjugates. Now, if we see the different functions of carbohydrate, then it can be used as the energy sources. During metabolism, the animal break down this carbohydrate to carbon dioxide and water vapor. Monosaccharides and disaccharides break down quickly and provide a quick energy sources, and starches takes a longer time to metabolize, but the end products are the glucose moieties. Human being cannot break down this cellulose as because they do not have the enzyme which can degrade the cellulose molecule. That means we do not have cellulase enzyme which is present in any type that cattle. They lack the appropriate enzyme which cannot break the beta-1,4 glycosidic linkage which is there in the basic skeletal structure of cellulose molecule. Now, if we see the other function of carbohydrate, then it is acting as a storage molecule. As I have told you that glycogen is the storage material where it is getting stored in the liver. Now, the main storage polysaccharides are starch and glycogen. Starch in case of plant and glycogen in case of the animal system. Now, when the body has the high amount of glucose, then the pancreatic release the insulin. The body can sense that body has got the system, the cell is having higher concentration of cellulose molecule. Then what this insulin is doing, that pancreas releases this insulin and it converts this glucose to glycogen, and this glycogen is getting stored in liver. When glucose concentration is low, suppose body is in need, in summer, I think many of you are sweating and sometimes we are feeling restless, and if we take immediate glucose water, then we get some energy. What does it mean? That means body was in need. In summer, when a person is sweating, we are giving that glucose water. So, when that body is in need of glucose, that means glucose concentration in the cell is low, then the hormone glucagon converts this glycogen back to glucose. And glycogen is the primary energy reserve in the human being. The metabolism of glucose provides the energy necessary for our body to function and carry out the daily activities. When it is broken down into glucose and oxidized, ultimately carbon dioxide and water is released through the cellular respiratory process. So, this is the major function, the storage function of carbohydrate. Now, if we see the function of carbohydrate as the structural stability, then we can find that cellulose is present in every plant cell, and in the plant cell this cellulose is giving a strong support. That means it has got the fiber like structure which is excellent building material of the plant. Now, when I have already told that cow, the cattle, they have this enzyme called cellulase, which is absent in case of human being, and that is the reason why we sometimes take green vegetable which act as an roughage for the system. If we consider chitin, I have told you this chitin is the structural component which is present in most of the this exoskeletal structure of the insect. Chitins is a leather-like structure, and it is the substance that eventually gives the hardness when it is shed. Chitin is often used in medicine for structure because it is both strong and flexible, but is also decomposed over time. Now, if we see the carbohydrate as a precursor molecule, then we can find that carbohydrate, that ribose or deoxyribose, they are the basic skeletal structure for DNA and RNA. The carbon skeleton of carbohydrate serves as the raw material for the synthesis of other small organic molecule such as amino acids and fatty acid. Disaccharide provide the building material for structure and protect the cell or whole organisms also. Now, when we are talking about the different structure and function of this carbohydrate, the polysaccharides are centrally involved in cell-cell recognition. How one type of cell is distinguished from another type of cell can also be possible because of these carbohydrate moieties. Some polysaccharide chain covalently bound by the glycosidic linkage to the hydroxyl group of the protein, which is called the glycoprotein, and act as the biochemical marker on the cell surface and also determines the blood group. Now, when we are talking about this carbohydrate as a vaccine, now protein, carbohydrate and lipid, as I have mentioned that these are the three macromolecules which are found on the cell surface of the pathogens too, are isolated and used as a vaccine. Carbohydrate requires the conjugated glycoprotein in order to be the immunogenic. Carbohydrate vaccine can be made naturally by isolating the carbohydrate from the surface of the pathogen or by biochemically or chemically synthesized, which is possible in the laboratory. Synthetic carbohydrate vaccine relative to their natural analog, which are essentially impure heterogeneous mixture, would be close to homogeneous mixture. They can be made via medicinal chemistry technique or by multi-antigenic. Thus, a single carbohydrate vaccine moiety could be synthesized to include the several antigens. This would prime the adaptive immune system to launch a multi-pronged attack, which would be more effective against the disease-causing organism. Carbohydrates are ubiquitous in nature and are basically just the sugar moieties. The toxicity of an ultra pure synthetic carbohydrate vaccine would theoretically be quite low. Carbohydrates are found on the surface of many types of cells, and thus vaccine for a variety of ailment, that is the malaria, cancer and HIV, exist in carbohydrate form. And this is all about the carbohydrate and its role in the biological system, in the living cell system. I think with this particular biomolecule, you will have some idea that how this micro as well as macromolecules are playing a significant role in the living system. So, with this today I am concluding. Thank you very much.